In the vast, sunburned landscapes of Australia, where eucalyptus forests stretch beyond the horizon and the soil carries the memory of ancient ecosystems, something extraordinary once took place. It began not with fire, nor flood, nor drought, but with insects. Hundreds of millions of them. Released deliberately, silently, and with a single purpose, to save an ecosystem on the brink of collapse. At first glance, the decision seemed almost absurd. To combat one ecological crisis by unleashing another living organism into an already fragile environment, an environment that had suffered deeply from past human interventions. Australia, after all, is no stranger to unintended consequences. From invasive rabbits to cane toads, history had shown that introducing new species could spiral into ecological disaster. Yet this time, scientists believed they had found a solution. A precise, calculated biological weapon. Tiny beetles, carefully selected, bred, and released in staggering numbers, over 500 million individuals, tasked with targeting a single enemy, invasive plants that were suffocating native ecosystems. But what followed was something no one had fully anticipated. Why would scientists risk such a massive ecological experiment? What made these beetles different from previous failed introductions? And how could something so small reshape an entire landscape? To understand this story, one must first understand the problem. Australia's ecosystems are among the most unique on Earth. Isolated for millions of years, they evolved in delicate balance. Native species, plants, animals, and microorganisms developed intricate relationships, forming highly specialized ecological networks. This biodiversity, however, is also highly vulnerable. When foreign species are introduced, intentionally or accidentally, they often lack natural predators. Without checks and balances, they spread rapidly, outcompeting native organisms and altering entire ecosystems. By the mid-20th century, large areas of Australia were under siege by invasive plant species. Among the most destructive were aquatic weeds like Solvinia molesta, a floating fern originally from South America. At first, it seemed harmless, an ornamental plant, admired for its dense, velvety leaves. But in Australia's waterways, it became a silent invader. Solvinia grew at an astonishing rate, capable of doubling its biomass in less than a week under optimal conditions. It formed thick mats across lakes and rivers, blocking sunlight from penetrating the water below. Oxygen levels plummeted. Native aquatic plants died. Fish suffocated. Entire freshwater ecosystems began to collapse. Wetlands that once teemed with life turned into stagnant, suffocating blankets of green. The environmental impact was severe. Water flow was disrupted. Irrigation systems clogged. Agricultural productivity declined. Local communities, dependent on these water sources, faced growing challenges. And traditional control methods were failing. Mechanical removal proved costly and inefficient. Herbicides posed risks to native species and water quality. Each attempt to control the spread seemed to bring new complications. It became clear that a different approach was needed. This is where the idea of biological control emerged. Rather than fighting nature with chemicals or machines, scientists sought to restore balance by introducing a natural predator, an organism that had co-evolved with the invasive plant in its native habitat. For Solvinia, that predator was a small, unassuming beetle, Certobagus salvinii. Barely a few millimeters in length, this weevil-like insect had a remarkable specialization. It fed exclusively on Solvinia. Its larvae burrowed into the plant's tissues, disrupting its structure and preventing it from reproducing effectively. In its native South American environment, this beetle kept Solvinia populations in check. The plant never dominated, never overwhelmed entire waterways. The ecosystem remained balanced. The question was whether this balance could be recreated in Australia. The stakes were high. Scientists conducted years of research before any release took place. They studied the beetle's feeding habits, reproductive cycle, and environmental requirements. They tested its behavior in controlled environments, ensuring that it would not shift to native plants or become invasive itself. The findings were promising. Certobagus salvinii showed an extraordinary level of host specificity. It depended entirely on Solvinia for survival. Without it, the beetle could not reproduce. This precision made it an ideal candidate for biological control. Still, skepticism remained. Australia's ecological history was filled with cautionary tales. Critics questioned whether the beetles could truly control the plant without unintended side effects. Others doubted whether such a small organism could make any meaningful impact against an invasive species that spread so aggressively. Yet the scale of the problem demanded bold action. And so, the project began. In a coordinated effort, scientists and environmental agencies initiated one of the largest biological control programs in history. Beetles were bred in controlled facilities, their populations carefully expanded to reach the necessary scale. Then, they were released. Not in small numbers, but in waves. Millions at a time. Across rivers, lakes, and wetlands. From Queensland to the Northern Territory, the beetles were introduced into infested waterways, their journey beginning quietly, almost invisibly. At first, nothing seemed to change. The thick mats of Solvinia remained, stretching across the water's surface like an unbroken green carpet. To the human eye, the ecosystem appeared unchanged. But beneath the surface, something had already begun. The beetles settled into their new environment. They fed, reproduced, and spread. Their larvae tunneled through the plant's tissues, weakening its structure from within. Days turned into weeks. Weeks into months. And slowly, almost imperceptibly, the transformation began. Small patches of the dense plant cover started to thin. Tiny gaps appeared in the once impenetrable mats. Sunlight, for the first time in years, reached the water below. Scientists monitoring the sites began to notice subtle shifts. The Solvinia plants showed signs of stress. Their leaves lost their buoyancy, turning from vibrant green to a dull brown. Waterlogged and weakened, they began to sink. As the plant cover thinned, oxygen levels in the water began to rise. And with it, life. Microscopic organisms, phytoplankton and algae, re-emerged, forming the base of the aquatic food chain. Insects returned. Small fish followed. It was the earliest sign of recovery. Yet, the process was not uniform. In some areas, the beetles thrived, rapidly reducing the Solvinia population. In others, progress was slower, influenced by temperature, water conditions, and local ecological factors. This variability fueled ongoing debate. Was the intervention truly effective? Or were these early changes temporary, destined to reverse over time? Scientists remained cautious. Ecosystems are complex, and early successes do not always translate into long-term stability. But as months passed, the evidence grew stronger. In several regions, the once dominant Solvinia mats began to collapse entirely. Large sections of waterways, previously choked with vegetation, opened up. Water began to flow freely again. Native plants, long suppressed, started to reappear along the edges. Birds returned to habitats they had abandoned years before. It was as if the ecosystem had been given a second chance. And yet, something unexpected was unfolding. Because as the Solvinia declined, the ecosystem did not simply return to its original state. It began to change in new and unpredictable ways. The removal of such a dominant species created opportunities, ecological niches that had been unavailable for years. Other plant species, both native and non-native, began to expand into these spaces. Some areas saw a resurgence of biodiversity. Others experienced the emergence of entirely new ecological dynamics. The beetles had done their job. But the story was far from over. Because what followed would challenge long-held assumptions about ecological restoration, and reveal just how complex and unpredictable nature truly is. At first, the changes appeared promising, almost textbook in their progression. As the dense mats of Solvinia collapsed under the relentless pressure of Certobagus salvinii, sunlight returned to the water's surface, penetrating depths that had remained in darkness for years. Oxygen levels continued to rise, restoring a critical balance within the aquatic ecosystem. But nature rarely operates in straight lines. Instead, it unfolds in layers, each shift triggering another, each recovery revealing new complexities. With the decline of Solvinia, one of the most immediate transformations occurred at the microscopic level. Phytoplankton populations surged, feeding on the newly available sunlight and nutrients released from decomposing plant matter. This bloom formed the foundation for a cascading ecological response. Zooplankton followed. Then aquatic insects. And soon after, fish populations began to recover in measurable numbers. In regions of northern Australia, researchers recorded increases in native fish density by over 300% within just 2 years of beetle introduction. Species that had nearly vanished from affected waterways, small-bodied fish adapted to low-flow environments, reappeared in sampling surveys. Bird life responded just as dramatically. Wetlands that had once been silent began to echo again with movement. Wading birds returned to forage in shallow waters. Waterfowl nested along the re-emerging vegetation lines. The restoration of open water created feeding grounds that had been absent for nearly a decade. From a distance, the transformation seemed nothing short of remarkable. An ecosystem, once suffocated, now breathing again. Yet beneath this recovery lay a more complicated reality. Because the collapse of Slovenia did not simply restore the past, it reshaped the future. As the invasive fern disappeared, it left behind vast quantities of organic material. This decomposing biomass altered nutrient cycles within the water. Nitrogen and phosphorus levels fluctuated, sometimes unpredictably, creating conditions that favored rapid growth of other plant species. In several locations, scientists observed the rise of secondary invasions. Water hyacinth, another aggressive aquatic plant, began to colonize newly opened areas. Though less dominant than Slovenia, it posed its own ecological challenges, capable of forming dense clusters that could once again restrict water flow. In other regions, native plants surged forward, reclaiming territory that had been suppressed for years. Species of submerged grasses and emergent reeds expanded rapidly, stabilizing sediment and providing habitat for fish and invertebrates. This divergence highlighted a crucial principle of ecological restoration: removing a dominant species does not guarantee a return to the original state. Instead, it creates a moment of opportunity, a window in which multiple outcomes are possible. And those outcomes depend on countless variables: climate conditions, existing biodiversity, water chemistry, and even chance. Scientists began to track these variations closely. Long-term monitoring programs were established across multiple regions, collecting data on water quality, species diversity, and ecosystem function. The goal was not only to measure success, but to understand the deeper dynamics at play. What they found challenged earlier assumptions. In many areas, biodiversity did not simply return, it exceeded previous levels. Freed from the overwhelming dominance of Slovenia, ecosystems diversified. A greater variety of plant species coexisted. Insect populations became more complex. Food webs expanded, supporting a wider range of organisms. This phenomenon, known as ecological release, occurs when the removal of a dominant competitor allows other species to flourish. But it also came with risks. Some ecosystems became more variable, less predictable. Seasonal changes had a greater impact on species composition. In certain wetlands, periodic blooms of algae occurred, driven by nutrient imbalances left in the wake of Slovenia's collapse. These fluctuations raised new questions. Was the ecosystem truly stable? Or was it transitioning into an entirely different state? The answer, it seemed, was both. In ecological terms, stability does not always mean returning to the past. It can also mean establishing a new equilibrium, one that differs from historical conditions, but still supports biodiversity and ecological function. This realization marked a turning point in how scientists viewed the project. The introduction of 500 million beetles had not simply solved a problem. It had initiated a transformation, one that extended far beyond the original goal. Over time, the success of the Slovenia control program gained international attention. Ecologists from around the world studied the Australian case, analyzing its methods, outcomes, and implications. It became one of the most cited examples of effective biological control in modern environmental science. Unlike previous introductions, such as the infamous spread of the cane toad, this intervention demonstrated that, under the right conditions, biological control could be both precise and sustainable. Key to this success was the specificity of Certobacus salvinii. Its exclusive dependence on Slovenia ensured that its population remained self-limiting. As the invasive plant declined, so too did the beetle's numbers. This natural feedback loop prevented the beetle from becoming a new ecological threat. It was a rare example of balance achieved through intervention. Yet, even as the project was celebrated, scientists remained cautious.